Hi, my name is Jessica Emery. The topic of my TAB is DNA in the criminal justice system. Deoxyribonucleic acid is a long molecule found in the cellular nuclei of living organisms. In the past decade, DNA has become an imperative tool in the criminal justice system. Federal and state courts have been increasingly accepting DNA evidence as admissible. The first state appellate court that admitted DNA as evidence was in 1988 in the Andrews versus Florida case. This was also the first major federal court decision to uphold DNA admissions in Jacobs. In Andrew versus Florida, the appellant wanted review of his convictions for aggravated battery, sexual battery, and armed burglary of a residence from the circuit court for Orange County, Florida. The court held that genetic fingerprint evidence by which strands of coding found in the genetic molecule of deoxyribonucleic acid were compared for the purpose of identifying the perpetrator of a crime was admissible. The next section of my TAB is identification in the criminal justice system. In several cases, DNA profiles can be obtained from bodily samples that became separate from a human body. Modern-day profiling techniques can generally be used on such tiny samples as the root of hair, cigarette butt, a blood stain, skin cells from clothing, or 3 micrograms of semen from a vaginal swab. Investigators become interested when such samples become separate from a person's body, which is usually either a victim or an offender at the time of the crime. The most vital use of DNA identification by criminal investigators is to compare a profile who is believed to be a perpetrator with the known person's profile. For example, DNA sample could come from semen in rape victim's vagina, blood, hair, or skin cells at the crime scene, or the victim's body, as I mentioned before. The next section of my TAB is identification in trial. In this article, the authors state when presenting DNA identification evidence in a criminal trial, it can be difficult for several reasons. First, DNA identification evidence compromises a number of facts, including the circumstances in which the relevant body samples were obtained, their secure transportation to a laboratory, their analysis, and the detection of recording of DNA profiles. There is need to prove all these facts, which the defendant might not be in agreement. Defendants are not usually willing to agree, which result in a long and complex proceeding and fact-finding. In addition, evidence law requires opinions about forensic interpretation to be presented by an expert based on training, study, or experience that substantially supports their opinion. Interpretation of DNA evidence requires expertise from various fields, notably genetics, statistics, laboratory technique, and crime scene analysis. On different occasions, Australian courts have allowed a person qualified in a single field to present an opinion based on several fields. Also, there's a controversy about how DNA evidence ought to be presented in a way that is both accurate and intelligible to people without scientific background, especially the jurors. Jurors that deliberate on a verdict must consider the DNA evidence along with all other evidence in the trial in deciding whether the defendant was proven guilty beyond a reasonable doubt. The next section of my TAB, I discuss a person that was exonerated due to DNA. Another case was the conviction of Dr. Crippen. Although this man didn't have the chance to be exonerated, forensic evidence accentuated proved his innocence. Dr. Harley Crippen was accused and convicted of murdering his wife in London in 1910. The reason for the conviction was microscopic analysis of remains found in Crippen's coal cellar, which were identified as Cora Crippen, his wife, based on a scar they said she had. As a result, Dr. Crippen was hanged, always proclaiming his innocence the whole time. Based on genealogical and mitochondrial DNA research, the tissue on the pathology slide that was tested later on that was used to convict Crippen was not Cora Crippen. It was actually the tissue of a male. The next section of my TAB, I discuss criminals that were convicted due to DNA evidence. The California Supreme Court made a decision in People v. Robinson, which was a case where a defendant was convicted of rape for an incident that occurred in 1994. The DNA evidence gathered at the crime scene provided convincing evidence that he was the perpetrator. Besides this evidence, the case was different from an important reason that was the authorities was the reason was the authorities did not locate Robinson until after the 6-year statute of limitation had ran out on the crime. The arrest warrant was just issued days before the statute had expired, but it did not contain Robinson's name or physical description. Instead, it only had a DNA profile created by the California Department of Justice. Robinson was found by traditional police procedure. He was found after computer. He also was found after computer matched the DNA profile from the arrest warrant with DNA collected after Robinson had been arrested for a second time. The next section of my TAB, one of my articles I discussed, was appropriate safeguards that exist to protect the DNA from misuse. The journal article focuses on the existing safeguards that are in place to protect the DNA samples from misuse. The main issue that arises with retaining the DNA sample is a person's right to privacy and civil liberties. According to Herkiman, the author Herkiman, there are legislative guidelines for DNA databases that are implemented to assist states with enacted DNA database statutes. There are specific provisions recommended to protect citizens' rights to privacy and protection of our civil liberties. In my TAB, I mentioned 4 of these recommended provisions, but for the purpose of the podcast, I only discuss one. DNA records that directly relate to the identification of individuals should be collected or stored for the purpose of obtaining information about physical characteristics, traits, or predisposition for disease, and should not serve any purpose other than to assist in identification of a suspect. The next article I cover is fairness. Over the past 20 years, DNA evidence has increased tremendously since 1989. The authors explain that almost every time DNA is presented in a case by one single expert, there's usually a conviction. Archival research reviewed that juries were 23 times more likely to vote guilty in homicide cases and 33 times more likely to vote guilty in sexual cases when DNA was admitted. According to the authors, the safety of these verdicts may be compromised by widespread misconceptions about the infallibility of DNA evidence by jurors. Field studies, interviews with actual jurors, and jury simulation confirm that individual jurors struggle to understand and apply the statistical information conveyed by forensic experts about the likelihood that DNA match occurred randomly or by chance. The problem with misunderstood evidence is that it conflicts with justice. Post-trial interviews of jurors who served on 6-month trials in New South Wales disclosed that jurors who admitted difficulty understanding DNA expert evidence and testimony, but still they convicted. To minimize biasness, they believe it was crucial jurors understood the experts. As a result, a study was conducted. The purpose of the study was to identify the factors that improve jury understanding and the use of evidence that links a suspect to a crime. The results of the study found pre-trial DNA knowledge in the 5 groups were low. There were no significant differences between the groups. After the exposure to DNA expert testimony, DNA knowledge increased significantly to 59%. The goal of this article was to illustrate the importance that jurors understand the DNA evidence. One part of my paper also discussed different ways that DNA is obtained. The article Recovery of DNA from Latent Blood After Identification by Fluorescein focuses on the recovery of blood by using fluorescein. The authors state that luminol is used in the field investigation to detect latent blood. However, luminol has the capability of destroying DNA evidence. Therefore, fluorescein is the alternative to luminol for detecting latent blood at a crime scene because it does not destroy DNA evidence. Violent crimes usually go unsolved because of the lack of evidence leading to a perpetrator. As a result, any evidence found at the crime scene is important to solving a crime. Violent crimes often leave latent evidence like DNA at the crime scene. In the United States, 13 short, tandem repeats low-count DNA strands are employed by the FBI for placing all felons in a national database, which is called CODIS, which I also discussed in my paper, and analyzing biospecimens found at the crime scene. Another section of my paper, I discussed the use of adhesive tape when obtaining DNA. The usage of adhesive tape for recovery of DNA from crime scene items is a crucial tool to obtaining DNA evidence. Authors also emphasize that selecting the appropriate method of collection of biological material from these crime scene objects can be crucial to obtain a DNA profile. When conducting this process, the 3 methods that are usually used are cutting, swabbing, and taping. The tape sampling works well because it allows the collection of a possible highly informative source of DNA, shed epithelial cells, and from selected areas on crime scene items, such as inside fingers or a glove. The authors continue when collecting biological material taping, it reduces co-sampling of known PCR inhibitors such as clothing ties. The authors state it is important to choose the correct choice of tape for crime scene item sampling because not all tapes are appropriate for biological trace of evidence collection and DNA extraction. The authors present 3 cases in which taping was used as the method of sampling of different crime scene items. They collect the epithelial cells from acknowledged areas of high friction between the body and the warm garment or the handle object. In 2 cases, they found selecting the tape of different areas of the same item, a gun and a glove, showed 2 different identical DNA profiles. The different cases are presented which show the usefulness of adhesive sampling of crime items. Finally, they mention the advantages of the tape collection technique and guidelines for preferred areas of tape sampling on several casework items were presented. The authors give a few examples of areas of sampling, and they are, for robbery or ski mask, it is around the inside corners of the mouth, the area of the nostrils, and below the eye holes. The eye holes shed a large number of epithelial cells. For gloves, the inside of the tips and the base of the fingers, and at the base of the palm. For socks that have been worn or used as gloves, it is the inside of the heel and the toe area. For guns on the slide, usually automatics, the trigger and the trigger guard. The hammer is usually the handle. The next part of my TAB, I discussed the value of DNA material. This article concentrates on the significance of DNA material obtained from crime scenes. Excuse me. The authors explain that DNA evidence is collected routinely from crime scenes for different offenses, and its timely process is recognized as an important component in solving a crime. The article reviewed a study which examined a different aspect of DNA process and how it can be used to solve a crime when there is a match. It showed the offenses where a DNA match was obtained. They state there are many predictors that influence the outcome of a DNA match. The analysis of the study consisted of 1,500 samples of DNA material recovered from the property crime offenses of residential burglary, commercial burglary, and a motor vehicle theft in North Hampshire, UL, during 2006. The results revealed that saliva and cigarettes was their main source of DNA recovered. Blood, cellular DNA, and chewing gum consisted of 63% of the samples. The study also measured the process of the DNA samples, its conversion to DNA profiles, and then how it is placed in the UK National DNA Database. Also, the process of a profile and obtaining a match in which crime scene examiners recovered DNA samples of different sources, the location of DNA obtained from the crime scene, and its mobility. It also found that the DNA material recovered illustrated several predictors besides the time factor, which really influences conversion to the DNA profile. According to the authors, the most important predictor was found to be the crime scene examiner's accreditation with offense types. DNA sample condition was relevant as well. A similar study of DNA samples profile showed a match with an offender on the UK National DNA Database. There was no significance with any of the predictors that were considered. In conclusion, my technology application brief illustrates the wider use of DNA in the criminal justice system. The use of DNA in the criminal justice system goes as far back as to the 1980s. Since then, it has exonerated more than 100 criminals. Prior to DNA, several criminals were wrongfully accused and some were even executed. DNA has also assisted in convicting several criminals as well. Ensuring the jury fully comprehends the expert's testimonies is crucial in a criminal trial. A study showed that most jurors convicted without even understanding the usage of DNA in its discovery. Also, there are many ways to obtain DNA through a person's There's also ways to obtain DNA through a person or a garment. Forensic scientists obtain DNA through blood, saliva, dental, hair, semen, urine, and body fluid, which I covered in my TAB. DNA seems to be more reliable than hearsay or eyewitness testimony. Each article provided a thorough explanation on how to retrieve this type of DNA. Dental identification is usually utilized when the body is badly disfigured or decomposed. This is my TAB podcast. Hope you enjoyed it. Thank you.